Hello, my students. Welcome to week 15 office hour. And this time, let's dive into the vocabulary words as well as the concepts of meiosis. So I will move the camera down. And let's first discuss about the chromosomes. So if I have this chromosome that has two arms, this would be, yes, this is the coiled up DNA, and those capital letters or segments in here represent alleles, like the A allele defines a character like the ear shape, and here is a capital letter that represents the dominant allele of this character. This is color-coded. This is from the mother. The offspring has another set of chromosomes that came from his or her other parent, the father. Now, check these. The blue one contains also at the same place an allele that codes for the same character like ear shape, but it's a lowercase a. The shape of the ear at this point represents a recessive allele for that character. So the organism for that character, the ear shape, is heterozygous, contains a capital and a lowercase letter. Now, these chromosomes, we did not mention that in mitosis when we dealt with chromosomes, but they are homologous chromosomes. Homologous means that they have alleles for specific characters at specific locations, but they don't have to be the same. For example, the orange one has a dominant allele for that, and the blue one has a recessive, but they code for the same character like ear shape. For example, the A. The E codes for a different character like the shape of a pinky, for example. So they are homologous. We discussed that the exact copy of the same chromosome is called the sister chromatids when they got attached, and when they separate during mitosis, the sister chromatids separate in anaphase and they move towards the opposite end of the cells. So for mitosis, it was like this one here, and that was here in metaphase, and both of the chromosomes, sister chromatids, started to separate from each other, and they formed the two cells, exactly the same cells. Now, at this point, we will discuss a little bit different stuff, the meiosis. So we go with one chromosome from the father, another from the mother, but we will do 2n equals 4 at this point. I might just move it a little bit back so you will see it a little bit more. And also, I have another set of chromosomes like this. So 2n equals 4. 2n, because it's diploid, I have 1 and 2 of the similar chromosomes, not exactly the same, but they are homologous, and they are homologous with each other, these two too. So I have 1, 2, 3, 4 chromosomes. We are in G1 phase. This is going to go through meiosis it's a spermatogonium, spermatogonium or ogonium, a diploid sex cell that will start to go through meiosis and forms haploid cells. But before we just go, every cell go through cell cycle and we hit the S phase. What happens in S phase? The duplication of the chromosomes. We have exact copy of those, exact copy of that chromosome, exact copy sister chromatids for that chromosome, and the sister chromatid for that chromosome. So now we still have 1, 2, 3, 4 chromosomes. They are same, but it counts as 1 chromosome, or it counts as 2 chromatids. In here we have 2 pairs. This pairs with that, and that pairs with that. Chromosomes, 2 pairs of chromosomes, or 4 chromosomes, or 8 chromatids. These are super important to understand the meanings of the words. Okay, so S2, S phase, G2 comes, and now we are in meiosis prophase. The prophase contains the special event, the crossing over event, and I would like to demonstrate the crossing over at this point. I just have the tape and a pair of scissors. So at this point, the homologous chromosomes get together. So this goes together and they form a tetrade. They on top of each other, top of each other. Okay, so we have 2 tetrades. What is happening in here, I tried to show it to you guys, like one of the chromosomes end part got snipped off and will change place with its homologous pair. Not with the sisters, because it would make no difference. But I would tape them all together like this. Okay, this is one crossing over. And we will just check. I take off the top of this and the top of that. And make the crossing over in here too. And the crossing over goes here. Good. And also now we can just also make another crossing over. For example, this R is still wanna cross over with this R segment. And let's finish the crossing over. Oh, it doesn't want to come up. Okay, good. And this one crosses over with that. Okay, so they went through crossing over. We can do the same stuff with the second tetrade. Goes with this, goes with this. The homologous pairs start to cross over. And with this and that. One that did not go through with this. I will snip off a bit of this too. So mix and match, right how it goes. And I think that is enough for crossing over. Makes a genetic variation with time. So we have now the chromosomes. They line up in the midline. No, 4 of them goes together. So the homologous chromosomes line up in the middle in metaphase 1. So think about when it was mitosis, only 2 chromosomes were in a midline, but in here, pairs, 1 pair of chromosomes will go in the line. At this point, they will start. I try to make sure that the red that is very close to the centromere will go with the red, and the green goes with the green. The blue goes with the blue. So they separate from each other. So these are homologous chromosomes, and they separate from each other. Okay, the homologous separates. Now they can separate like this. Now I'm just introducing another word, the independent assortment, but they can separate like that too. So that independent from the separation of the first pair. They can separate like this, or they can separate like that. That's the independent assortment that relates to and contributes to genetic diversity too. Let's do this way, doesn't matter. So now we are having 1 cell after the cell first phase of meiosis, and another cell. After this, the space is quite small in here. So what we will have, the second division is the sister chromatids will separate from each other, okay? And I will put next to each other. So this will be 1 cell, form a cell that is haploid, has only 1 set of alleles like from A and B and D and all of them. And we have another cell. And the second cell that separated during meiosis 1 will separate again. And now we have another cell and another cell. So we have 4 cells, 1, 2, 3, and 4. Check that. All of the cells have different genetic material. None of them has blue, lowercase a, b, d, c, and uppercase F, G, H. See, it has lowercase a, b, but it has an uppercase D. And this one doesn't have lowercase a, b at all. So all of the cells are different. And all of the cells would be different also from that scenario if I would play through the independent assortment with the second pair of chromosomes too. So what else? Okay, good. Now, there's a thinking question on like the donkey and the horse. So when the donkey is one of the parent and the horse is another one, they produce a mule, and the mule is sterile. And the question is, why is the mule sterile? So the question, so the donkey has 62 homologous so they 62 chromosomes, so it's 31 pairs, and the horse has 64 chromosomes. Now 62 and 64, the mule has 63 homologous chromosomes, but the 63 cannot really line up evenly in the middle. That's why the mule is sterile. The cell cannot go through meiosis because 63 chromosomes cannot line up in pairs in the metaphysio line during meiosis. One of them would miss its homologous pair, so they cannot separate from each other. Well, not like this, but they would just wait for, think about like this. Well, actually, you know, I already just did the crossing over, so they cannot really just have another additional pair for crossing over. I guess that's all about meiosis at this point. So think about the crossing over in prophase 1, as well as the independent assortment in metaphase 1, as well as the fertilization of a specific sperm, very unique in genetic makeup, will fertilize a genetically unique egg that's also very random. So that's why we have huge genetic variation that occurs in sexual reproduction. I hope that's all. Have a good one.